My name is Tim Clark. I'm a tenured associate professor in public health at the University of Virginia. And my colleague, close colleague, Sadnan Al-Munir, is also on this call. He is a research scientist in our team and has been extremely highly involved with this work. So I'm going to talk about the FAIRSCAPE Digital Commons for Ethical AI. And it's focused on applications in translational medicine. And those have special requirements, which I'll start to talk about now. So let's see. Does this advance? Okay. So we're going to talk about AI readiness requirements. We're going to talk about the kind of collaborative agile development we've done on this framework over the last 5 years with actual research programs in both functional genomics and clinical medicine. The results, I'm going to talk about how FAIRSCAPE processing works, the architecture, what it provides, our current work, how we evaluate AI readiness and what we're working on in that, and then some conclusions. At the end, I also provide download links for some data sets and the software and documentation. So AI explainability, I hope people are familiar with that concept. It simply means that for many applications in AI, it's not obvious how the results were obtained. You can have certain applications like decision trees that are inherently explainable, but as soon as you start to get into deep learning at all, you have to have uh, explainability of how the results were achieved. And especially in biomedical applications, there are often rather elaborate pre-processing steps that can affect potential bias in the applications and so forth. So and biomedical applications in the laboratory simply have the ethical requirement of being good science and having valid conclusions. In the clinic, it's a little bit more impactful because it can affect people's lives, life or death situations sometimes, when you talk about diagnosing a cancer, for example. So there's a requirement that in biomedical data sets that are destined for AI analytics, that you provide what is called pre-model explainability. Pre-model means before you do the model instantiation, the model development and uh, instantiation and processing. There are other issues that come into play with in-model XAI and post-model XAI that I'm not going to talk about much here unless people ask questions about it. We're concerned with pre-model explainability or AI readiness. And the way we do this, just in a nutshell, is we provide very, very rich metadata in RO crates with provenance graphs, semantics, statistics, ethical characterization of the data sets, computability, a number of other issues. And we support all the NIH, the US National Institutes of Health Bridge2AI AI Readiness criteria, which we helped develop, but it was developed across the Bridge2AI consortium, these criteria. It's a very big consortium with 40 institutions, 130 million US dollars in funding, and it's in its fourth, going into its fourth year now. So we support currently functional genomics and clinical programs, actual real programs in these areas. So this is a graphic of the criteria. And I think if you look in this little circle, they start with fairness, and we're all, I hope, familiar with the FAIR data principles: findable, accessible, interoperable, and reusable. Let's talk about these as a clockwise progression. Fairness requires provenance, but in our view, the kind of provenance that's required for AI readiness is deep provenance, and that's not necessarily well fleshed out in the FAIR principles. Furthermore, we require semantic, statistical, standards compliance, biases, and data quality characterization, what's called pre-model explainability. Is this data fit for purpose, verifiable from its source, and data documentation templates, aka data sheets. There's a famous or relatively famous ACM communications paper by Timnit Gebru et al. From 2021 that describes uh, data sheets in a conceptual way. There have been different approaches to this, but the basic idea is that you have a very rich description of the data sets. We add ethics. In developing these criteria, there were professional biomedical data ethicists on the team. Sustainability. This is a bridge to AI requirement, but it's also, if you think about it, fair principle that if data goes away, it's not accessible anymore. So we require that it's persistent, appropriate to the domain, well governed, and associated with related kinds of data. And then lastly, computability. I'm going to note that many times in conversations with computer scientists that are really AI specialists, they start with this. They don't care about any of these things. They just want the data. Give me the data and I'll build a model that will compute certain things on it. And I say you cannot take biomedical data as a ground truth if you don't know where it came from, how it was processed, and so forth. You cannot. Right? And happy to debate that with anybody who objects. But the computer scientists are really interested in their models and model implementation and so forth. And not to say that we're not computer scientists, but the sort of pure theoretical people tend to, and I hope I'm not slandering anybody, this is a limited sample set, but they tend to just think, give me the data and I'll work from there. Possibly because this information hasn't been available. So what methodology did we use to build FAIRSCAPE? Well, we developed it in collaboration with AI machine learning research teams in predictive clinical medicine and functional genomics. So we started off working on a predictive application at the University of Virginia on infants, and I'll talk about that a little later. And then built an earlier version of this software, which we use for preliminary data in applying to be funded under Bridge to AI, and we got funding. We've gotten a fair amount of it. It's now we got a supplement about a year and a half ago to make it the program packaging standard for the whole Bridge to AI program, which consists of four grand challenges: functional genomics, clinical care medicine, voice waveforms as a predictor of health, and a final one on diabetes. And we support very large data sets in multiple format, plus extensive AI readiness metadata in RO-Crate packages, which means that we use schema.org metadata. So let's talk about the first clinical predictive monitoring program that we worked on in the NICU. That's a neonatal intensive care unit at the University of Virginia. Our colleagues in that unit, Randall Morman, who recently retired, Karen Fairchild, Bryn Sullivan, and others have been collecting continuous monitoring data on these babies who often are, they're premature infants that are often in not such great shape. And they're put in incubators. They have all these leads on them to check their oxygen saturation, heart rate, ECGs, sometimes EEGs, and other electronic signals as continuous waveforms. And we did a project to take the 10 years of continuous monitoring data, and we took a big collection of waveform analytic algorithms out of the literature, not necessarily from biomedical applications, but biomedicine, stocks, all kinds of things that have continuous signals. And we applied them with a lot of different parameter sets in basically almost 5,000 different combinations. And the goal was to predict an adverse event. I'm going to say adverse event, but really the adverse event we were trying to predict was that the baby die within the next 7 days. And we could do this prediction, and we actually found a really good set of algorithms to do this. And this work has continued, and we now work with Dr. Sullivan and Dr. Fairchild and their teams at UVA, Birmingham, Alabama, Columbia University, and Washington University St. Louis in a collaborative project on doing different kinds of analysis on these neonatal babies. Then in the Bridge2AI program, what we're working with, we're trying to produce hierarchical cell maps of the internal protein subunit arrangement and rearrangement from data taken from different kinds of cell lines that are perturbed. So breast cancer cell lines that are treated and untreated, so treated with vorinostat and paclitaxel. Also, human-induced pluripotent stem cells, iPSCs, that are genomically perturbed across all the genome, which is really an amazing project out of Prashant Mali's lab at UCSD. And what is done with these data sets, so we have protein-protein interaction, protein localization, subcellular imaging, and the perturb-seq data from single cells on the same cell lines, and they're processed through a rather elaborate pipeline that generates embeddings, machine learning embeddings, actually AI, deep neural network generated embeddings off of these these data, combines the embeddings and does hierarchical community detection at multiple levels to get an architecture of the cells. And the idea behind this is to be able to use these cell maps in further AI applications. So the idea being that if you want to do additional computations for different kinds of cell lines, you can use the same approach in the same pipeline, or just take the cell maps that we produced and take a look and see what perturbations seem to affect in terms of the actual protein subunits in the cell. And this is an example protein subunit hierarchy from an earlier experiment on the U2OS cell line. And you can see that you start at the highest level, everything in the cell, and then it starts going down to very small protein interaction groups. And those change when you perturb, you either genetically or genomically perturb the cells, or you perturb them biochemically with therapeutic treatments. And that is helpful and insightful in terms of understanding what's going on in the cells. So the point I'm trying to make here is that we didn't just sort of make up a an architecture for our system based on nothing. We based it on, well, there's earlier theory behind it, and I'll get into that later, but it was based on applying earlier theoretical lessons and architectural experience to support real programs. And so we did it in agile development. And I'd say we've probably, Sadnan could correct me if I've misspoken, but I'd say we've probably done major refactoring on this system at least 5 times, maybe more. This is the processing that we do for the cell map. So you can see that where it says data acquisition pipelines, those are the different inputs, lab inputs to this system. And these are at different labs. These are at the IF microscopy is done in Emma Lundberg's lab at Stanford, the APMS, and also SECMS protein-protein interaction data comes from the Krogan lab at UCSF, and the RNA-seq data comes from Prashant Mali's lab at UCSD. And then the tools pipeline is run in Trey Ideker's lab at UCSD. And then the FAIR integration is run here at the University of Virginia. And what we do remotely is we have remote client applications that produce RO crate packages for the major steps in processing. Those are compressed and uploaded to the FAIRSCAPE server, where we produce persistent identifiers and store the data and make the graphical provenance traces available and so forth. So this is the architecture of FAIRSCAPE. The FAIRSCAPE, we have 2 clients right now. We have a command line client. This is all in Python, by the way. Originally, FAIRSCAPE was not all in Python, and I'll get into that later. But the command line client is in Python, and it's callable from the command line or as Python functions. We have a GUI client. I should say it's an Electron app, so it actually uses JavaScript, so it's not all Python. But we produce these RO crates, and then they're uploaded through a REST API. The server has a web GUI where you can look at the data that's been processed, and because we take some pretty large data packages. It may take a while to upload. We have a non-blocking architecture, so when you do an upload, the data goes into a message broker and it's temporarily stored, and then you can get the next upload package, and these are just queued up. The metadata is stored in MongoDB, metadata plus permissions, by the way, stored in MongoDB. Sorry, have to turn this off. Okay. The data is in Minio, which is has S3 compliant buckets. So we can run this on a laptop at the UVA data center. We run it in a much larger data space on the UVA research computing system. We've also run it on Google Cloud and in Amazon. We don't run it in those cloud environments right now because our access to UVA data center is nominally is free, right? So they do run, it does run a Kubernetes environment. And we also have interfaces to the Harvard Dataverse and Figshare archives. We're grant funded, and as long as we can keep the grant money flowing, this system is going to be alive and kicking. But the requirements in Bridge2AI is that you have to pledge that your data is going to be available at least 5 years after the end of the program. And so we also have interfaces to these NIH generalist repositories for long-term archival. This is just an example. This is taken from our June 2025 data release of the human readable form of the RO-Crate metadata.json files, not including the schemas, for the release as a whole. We have data, we have multi-level RO-Crates. So we have release level and modality level, and then individual data set level. So you can see that a number of these things, if you wanted to go back to that diagram, that sort of wheel diagram I showed at the beginning, map back to that diagram. This is a screenshot of the provenance graph that we produce, that we generate in FAIRSCAPE. Is a visualization that we generate from the RCrate metadata.json files. And if you want to read this in chronological order, there it's right to left. So we track provenance back to the samples and instruments that produce the data. So samples and instruments, experiment that was run on the samples, the raw data set output, data set collections, where there is software involved in processing, the provenance graph links to the actual software in GitHub or Zenodo. And we also link to the computations and have descriptive information about what computations were run and when and where and with what parameters. Far left is the output data set. So we use the ARC scheme developed initially by John Kunze and others to provide PIDs for these entities. And the reason we use ARCs is that there's no fixed metadata scheme for ARCs as there is for, say, DataSite. And not to say that we don't use DOIs. We use DOIs when we output to Dataverse, and you'll see that later. But DOIs cost money to generate, and they also have a fixed schema for the metadata. So we wanted to go a little beyond that. As I said, we used schema.org metadata vocabulary, but we also combine that with evidence graph ontology, or EVI, which we developed by subclassing a lot of terms from PROV. You can see it in the dotted line rectangle, some of the base classes in PROV, W3C PROV. And we subclassed it and designed the subclasses to support different elements of biomedical data sets, including RO-Crates, including the graphs themselves, and so forth. This is under further development right now, and we'll tell you more about that later if you're interested. The fact that we're trying to comply with these AI readiness criteria from Bridge2AI means that you can do evaluations. And in our paper, the AI readiness for biomedical data paper cited down at the bottom, we did these evaluations for all the projects, both currently at the time we wrote the paper, and the current evaluation. Here's an example of one project that isn't completely AI ready in the blue lines, right? And it's great because you can then you can then say, okay, well, what do I have to do to fix this and get out to the orange lines? Um, the way we do this evaluation is we just we give people a one if they met one of these sub criteria and a zero if they didn't. And then, as you can see in Providence in this example, uh, we hit three-quarters of the criteria, so you get a 75, right? So this is the idea. It's a little, how can you it's not weighted, okay? It's not a weighted average, but it gives you something. And you can create these radar plots. Now, can we do automatic evaluation? We'd like to, and we are nearly complete with developing an algorithm to do this automatically, but there's a little gotcha here. Data documentation templates. If you remember, when I showed you the release level metadata, there's a lot of verbal description. And how do you know that somebody just didn't type in ASDFGHJKL into their verbal description to pass muster? Well, you don't. We're talking with our colleagues at Lawrence Berkeley Labs who do a lot of LLM work, and it's possible that we could do evaluations on these using LLMs. The other thing is that, of course, people that publish their data with a description that says ASDFGHJ are going to come in for some pretty nasty criticism from their colleagues. So that's another sort of inherent human evaluation. Data and software documentation is here. The time series analysis on NICU babies is here. The most recent release of cell maps for AI is there. And what are we doing currently? A lot. We're extending the RO-Crate packaging across all four Bridge2AI grant challenges, and we're tidying our conformance to RO-Crate 1.2. We have one issue that we have to resolve that requires some server changes. We do not have flat JSON LD, and the reason for that is we included the schema descriptions in the JSON LD to make it easier on the server to not have to retrieve separate files. But we will fix that probably within the next month. We have a collaboration with Lawrence Berkeley Lab and University of Colorado to generate data sheets with LLMs. And for the AI readiness evaluation, we have a collaboration with MIT that is just sort of getting off the ground to generate RO-Crate packaging for PhysioNet datasets. And we have a collaboration with colleagues at Yale to track in-model explainability as the models are developed. So in summary, FAIRSCAPE is evolving. It's a robust platform. It's used in practice in a couple of big collaborations for ethical AI readiness and biomedical data science, and we're working closely with colleagues to develop its capabilities. It's available under Apache 2.0. Initially started with MIT license. I don't know, Sadnan, if we have finished updating our license documentation, but we're switching over to Apache to make it more, let's say, amenable to collaborative development with other groups. And developing an ecosystem around these kinds of platforms is a large scale collaborative effort. And we'd like this work that we put a lot of effort in to be able to survive, at least as a starting point for other people's development. And so we welcome interested collaborators. And these are my acknowledgments. Teams from University of Virginia, UC San Diego, UC San Francisco, Stanford, and Yale. And a special shout out to Carol Goebel at Manchester, Marianne Martone at UCSD, and Randall Morman at recently retired emeritus professor of medicine at UVA, who helped us in many ways, with advice and consultation.